Gotta give it.
Forget about it. Hey yo, at the speed of sound, the world-renowned wax-works compound. So gather round and witness something that is rarely found.
The Database Building Blocks seminar series is filmed in front of a live studio audience. This program is made possible by Google.
All
right, let's get started. We're excited today to have Trent. He is formerly from Where True Technologies, where he built this Exon database system. But now he's also a software engineer at Meta. So he's going to talk about what he was building at Exon before. So again, as always, if you have any questions for Trent, please introduce yourself, interrupt at any time. That way Trent's talking to an audience and not to himself on Zoom for an hour. And feel free to do this at any time. So again, Trent, thank you so much for being here. The floor is yours. Go for it.
Awesome. Yeah. Thanks, Andy. So super excited to be here. Talk a little bit about Exon. So we'll talk a little bit more about Exon here in a minute. I guess I'll just try to do kind of a brief intro. So I follow maybe like somewhat of like the traditional archetype of sort of self-taught, and so sort of started out with sort of like Counter-Strike script kiddie, like very back in the day. And then went into accounting and finance and analytics, and so maybe a much more like traditional background for somebody who is working on like analytics or databases, or sort of the application of those to some problem. But then I got really lucky and ended up sort of falling into bio, and Exon is a database that is sort of purpose built for bioinformatics and sort of bioinformatic applications, and so kind of excited to talk a little bit about that. And so that's been basically the last 10 years or so prior to me joining Meta was in bio in some form, either working at a company called Zymergen doing sort of next generation sequencing analytics, or metagenomics discovery, which is basically a problem where you have a very big database of sequences and you have to search that database for often something as simply phrased as like, find me all the sequences that kind of look like this other sequence, because if it looks like this other sequence, it probably has a similar function to that other sequence. And we'll talk a little bit more about sort of those problems, how they play out in the database perspective. And then I live in sort of beautiful Shoreline, Washington, which is just north of Seattle and in the Pacific Northwest, and love it up here. I also do quite a bit of contribution to the Data Fusion project. So I helped from an administrative perspective adding scalar UDF supports for UTF-8 views. So folks who are familiar with the Data Fusion project know that sort of recently added and has been adding sort of improved support for UTF-8 view, which was exciting. That's also particularly exciting for bioinformatic applications, where so much of the work is about comparing strings and working with strings. And so if you can sort of use some of the niceties of UTF-8 view for, say, filtering strings and DNA sequences or protein sequences and so forth, that's really potentially exciting. Some sort of bound improvement, streaming blocks use it. So I'll talk a little bit more about this later, actually, in the context of how bioinformatic file formats tend to be compressed and indexed. But basically, most people don't think or don't know that you can often take a gzip file and concatenate them together into blocks. And so you might have a single file with multiple blocks, and that's how the indexing works a lot in these bioinformatic file formats. Added lots of docs, and then obviously everyone here is probably familiar with DuckDB, and they've done a lot to sort of push forward what people think is okay with sort of SQL as a language. And so I added support for basically count with no arguments and sort of negatives and split part and some of those other sort of smaller utilities. So have added quite a bit, or sort of been involved in the project quite a bit, which has been exciting. So before we get into my talk, I just want to give a little bit about sort of where Exon is in this talk, sort of in the context of the talks that have happened so far in this seminar series. So just briefly, we had the first talk, which was given by Andrew Lamb, and amongst a lot of things, he really focused on this, or he talked quite a bit about sort of like trait and extension-driven functionality, which is how Data Fusion allows people to add utility to it as a database. And so we're going to see a number of examples in this talk today about how I and via Exon have taken some of that capabilities and added bioinformatic capabilities into a database. Then we had some really exciting, sort of like the Spark Accelerator, I was thinking is great, Data Fusion Comet. And then the last two, sort of PrayDB and Spice AI, which are both, interesting, where you have applications, a lot of like text search and the PrayDB use case, and then some of this idea in Spice.ai about sort of data federation for acceleration. And some of this idea of sort of data federation is kind of tangentially related to Exon, in that a lot of bioinformatic data is stored as basically files. It's a very file-based science, and so we're going to talk a little bit about how I have sort of adapted bioinformatic or using like database file processing that you see, all over in these sort of like embedded OLAP databases to bioinformatics, and some of the sort of nuances and challenges there. I want to just give a kind of brief overview into sort of bioinformatics data, and then we'll map it on to what it looks like as an actual a database. So when you're doing like informatics or bioinformatics, if you read the sort of textbooks around it, you really break it out into three main phases. Sort of primary, secondary, and tertiary. Primary is really about getting the initial sort of biological signal, and whatever that may be. So base calling, for example, which is literally given some strand of DNA, you calling the individual bases on that DNA, say an A, a T, a C, or a G. Also, a lot of the instruments will then generate some QC scores, which are useful for filtering or other sort of quality control analysis, or what's becoming increasingly popular as a modality, which is imaging. So this is like getting the initial signal. Then you sort of move up into secondary, which is really around sort of like alignment, which I'll show an example of in a second, or sort of variant calling, which is say, okay, like, so-and-so has some disease. Is there some sort of genetic motivation, so to speak, for that disease? And really, do they have some genetic variant relative to most people that could be causing that disease? And then all the way on top of there is sort of tertiary analysis, which is sort of annotation, comparative analysis, which, folks who write a lot of SQL will do a lot of this work in their sort of day-to-day, where you have, I have some data set that I need to join with another data set that I need to join with another data set, ultimately filter that down and do some analysis. And that's tertiary analysis. And that's what a lot of sort of Exon can help with today, although I hope to, over time, sort of push it down to these other two layers. Not going to spend a lot of time on this. I just think it's kind of interesting to give folks a little bit of a sense for what's happening when like the primary level is, or the primary part of, say, base calling is happening. So this is this is a nanopore sequencer, and basically the sequences of DNA are running through this nanopore, and the pores can then be attuned to the different electrical signals that the nucleotides or amino acids give off. That's ultimately how the strands become sequenced. And so, it's sort of super fascinating. It's way outside the scope of this talk in terms of how that actually takes place, but I think it's useful to sort of like build up from there. So then once we have secondary, again, we talked a little bit about alignment and mapping. So we'll talk more about this as we go on. Variant calling, again, like I mentioned, is just sort of understanding variants in people's genomes relative, or any sort of sequence relative to say a reference. Quantification, and then assembly, which often happens if you have a lot of individual DNA reads and you need to somehow combine them into some sort of cogent larger genome or contiguous chunk of DNA, and do so via assembly. So I just want to show kind of an example of what that looks like. So this is alignment, and what we have here, and this is an awesome tool. I was lucky enough to work with the creator of HighGlass when I was at a company called Zymergen. And basically what we have here is a reference genome. So that's the genome on top. And then we have all of these individual horizontal lines are sequences or reads, and they are aligned back to the reference genome. And what we'll see when we zoom in here in a second is the sort of differences or the sort of variance, right? So like this is sort of how, if you imagine we have reads from the last slide that we saw running through that nanopore. What we have here is the sort of secondary analysis where we are then able to say or make some call, and there's some statistical analysis that goes into making this call that say in this position, we see an A, but it looks like a number of the reads are a G, and so that might be an actual like variant, which could be interesting, or everybody has tons of variants all the time, and there's something called sort of reference bias in terms of like, is the reference the best one for an individual, and sort of lots of questions that come up, but suffice it to say, this is the sort of next level after primary is, okay, can we get some sense of how a set of reads sort of aligns back to a genome, right? So this is sort of this is secondary. Tertiary, and we'll cover this a little bit more because it's kind of where databases can have the most immediate impact in bioinformatics is combining this data together. So in this case, we might have sort of VCF data, so this is like variant call data. We might have like demographic data and or sort of clinical data. And in a very simple case, we're taking this data, joining it together, and hoping to get some insight out of it to either understand what's wrong with an individual, or for work that I did, a lot of times it's trying to like either find improved proteins or find improved metabolites. It's been saying like to the scientists you work with, hey, these 15 are interesting for whatever reason. Let's continue the experimentation process with that. So one of the challenges in bioinformatics, and sort of, I would say a lot of these fields that have progressed relative to, or sort of in parallel with sort of software generally, is they tend, like bioinformatics especially, tends to be a very like file-based science. And so a lot of the outputs of these tools are just files. And some people do certainly work with databases, traditional databases, say, like your OLTPs of the world. But due to the nature of the work, where it's a lot of workflows, which we'll cover in a little bit, we're ended up dealing with a lot of different file types. And so Exon basically has a notion of a file type, and we more or less map on and have the capabilities to parse and turn all of these file types into database tables, or really using Data Fusion's table providers, such that we can parse these and run queries on them like anything else.
Trent, a quick question. I know you said you're going to focus on the tertiary structure, the tertiary part, but do you also store references to the secondary and primary if someone needs to go back to the original images to go and verify or rerun a pipeline from there to get to the tertiary? Is that outside the scope of the system, or does it get linked in some way?
Yeah, so that's a great question. That is outside of the scope, I would say, of like Exon as a sort of like maintaining the data lineage, so to speak, is if I'm sort of like kind of what you're referring to. There are tools, and I'll mention that in a second, that are sort of custom built for that. Maintaining lineage, like sort of as a workflow, but at least in Exon, there's not a notion of sort of like data lineage or like workflow lineage. But it's a great question, and definitely important for doing science in a reproducible way.
Got it. Thank you.
Yep. Yeah, thanks for the question.
Are these files, are they standardized across the industry, or is like every instrument spits out its own file format, or is everything like using HDF5? And how standardized are these things?
Yeah, that's a great question. So they're like pretty standardized. I would say it's like one of those things where like 95% of all of these files that we're looking at look the same. There's some sort of challenges on the margins. There's lots of file formats that are kicked out in other aspect of biology, that it is somewhat very sort of idiosyncratic to the vendor that generated the file, where they'll just kick out some text file, that text file will follow whatever format the vendor ascribes. But at least these file formats, which are pretty widely used in sort of bioinformatics, both sort of academia and industry, are relatively standardized.
So the format that we standardize, but like, are the semantics of what the values mean, is those like reasonably standardized as well?
To a point. So
Okay.
I'll mention a little bit with like DCF, sort of like a variant call format. So that's a file format that stores the schema in the header. And so you can sort of parse that header, and it has a notion of basically like an info field. And in this info field, there are standardized tags that are defined in a spec. So there is like a VCF spec which exists, which is sort of like maintained by a group. There's also the ability to sort of add extra tags or extra metadata to, like, sort of based on if you need it in your experiment. So yeah, there's a sort of core spec which is defined and followed pretty well, but there definitely is wiggle room for better or worse.
Thank you.
Yeah, thanks for the question. Okay, so let's talk a little bit, and I think let's look at this is great. The question a second ago was great about data lineage. So like, here's what you often see from like a workflow perspective for tertiary analysis, and some of these parts or many of these parts can be done inside Exon or can be sort of extended with Exon. So this is a traditional RNA-seq pipeline. I don't want to get into all of it, but suffice it to say, each one of these little stops in this subway chart are basically some CLI tool which comes out of academia, which is applied sort of like in succession to carry out the workflow. So you have like FASTQ. So FASTQ is a data format which comes out of NGS. They get concatenated together, then sort of infer the strandedness. You then do FASTQC, which is like quality control. And so you have these pipelines which are fairly standardized and produce the outputs, that the subsequent steps can use and process. And so that's great. And some of these, like I mentioned, can be done inside Exon. And some of them, it's like, okay, it's great we have this workflow when we did it, but that workflow doesn't necessarily allow you to answer kind of like the ad hoc questions which may come about, or do perform, say, analytics on top of this pipeline, which is necessary for your tool. So like, for example, say you wanted to, like BAM has this NH tag, and again, we'll talk a little bit more about the sort of schema on read here in a second. But you have this BAM file, which is basically an alignment file, and a BAI, which is like the associated index. So basically, yeah, like you see in databases, in bioinformatics, lots of file formats have indexes along with them, and it's basically two files that come together. So if you were to, for example, wanted to do like analysis on the edge of this workflow, this is something like a query that you could run in Exon, where you are basically selecting the number of alignments. That's this sort of NH tag, which you kind of need to know. And then from your BAM file, so this would be the actual like file on the disk. And then we'll talk a little bit about this here in a little bit, but yeah, basically supporting sort of like predicate push down into the index so that you can only select the alignments on chromosome 1 between 1000 and 2000 for your specific file. And so, yeah, a lot of the sort of bioinformatic tertiary analysis and sort of tertiary workflow is very CLI driven, but you're somewhat constrained by that CLI and its sort of inputs and outputs relative to, say, ad hoc analysis that you might want to do on the edges of that. Okay. So I want to talk a little bit about, so that's kind of like the high level, how we sort of get into tertiary, and then let's talk a little bit more about some of the idiosyncrasies of the data and really like how that'll map onto how Exon uses data fusion in particular to make it so we can take advantage or sort of natively understand these idiosyncrasies. So just want to give like a quick overview of a FASTA file. So this is the FASTA, and this I think is just a sort of a nice illustrative example of some of the challenges in bioinformatic data processing. Astute folks will know this sort of like constant discussion around or the sort of lore, so to speak, around why terminals tend to be 80 characters in width. It actually turns out that like FASTA files also were sort of shaped by that smaller monitors of years gone by. And so the typical format for a single sequence like this is basically 80 characters in length. And so you look at these sequences and these files on disk, and they're actually multiple row or multiple lines for a given sequence, where it's an 80 character length limit as like one line. So this is one sequence that's broken up across many lines. So you can imagine some of the challenges that come along with, say, parsing even something like simple like this, where you have a header, which is basically this little caret up to the first space. Anything after this first space is the description of the sequence, and then everything else is the actual sequence itself. And these are basically concatenated together multiple times to create a sequence file format in the FASTA, which is ubiquitous in bioinformatics. The next file format, which we'll talk a lot about, is VCF files. So I mentioned it a bit earlier. This is a file format that came out of the 1,000 Genomes project, if the folks have sort of heard of that, which is basically this like large-scale longitudinal study that has more or less followed individuals along for a very long time, trying to understand their health from like a clinical perspective, and then also understand their genomics to try to then make connections between their genomics and any sort of health outcomes that they could have. So this, I think, Andy, this gets to your sort of like question point a little bit earlier, where so this is what a VCF file looks like, and up in this top part with the sort of double pounds, we have the header. And so if we look at this, we basically see like we have in this info field, which is, you can see like DP equals 1, colon, I16 equals 1, 0, 0, 0, 26, and so forth. We have the fields and we have the numbers. So this could be, so like this first one is an indel, which stands for insertion or deletion. It's basically, it's a flag type and it's got its description. Or you have like QS, which is, so yes, this is sort of like auxiliary tag, and it's a float, and you could have many of them. And so you can see, or you have format, where you have, yeah, say like the genotype, which is number 1, and it's a string type. So really you have like the schema of the file is sort of like embedded in the file. Yeah. And so it's not like super complicated, but you can imagine that if you want to make a user interface to be able to run SQL against that, you have to say, okay, like how can I turn these, say, info fields into something that's like a struct or some other type that you could then run the select star, select info dot whatever against. And so this, and then I wanted to mention briefly, I'll talk about this more from the data fusion perspective here in a minute, but so indexing compression, which is something that, yeah, obviously happens in sort of ubiquitous from a database perspective. But the way this works for VCF files and a lot of other bioinformatic files is, say I sort of start out and I want to query some specific region, say like chromosome B from 1 to 1,000. The way that actually works is you have some block gzipped file and an associated index file. And that index file basically has a mapping from, say, the location in the genome, say chromosome 1 from position 0 to 225. And then it has the byte offsets within the block gzip file format that you want to query. So when you're going to run a query, you basically look up in the index the byte offsets that are associated with what you want, and then you sort of go back into the VCF file and select your particular blocks that you're interested in. And so when folks don't do this from like when they're using, say, like the command line tools, this is sort of hidden from them. But obviously, if we're going to build a database on top of this, which can run at or faster than the same speed as these tools, we have to sort of understand how this works. But this sort of indexing and compression is kind of common. It's in VCF files, it's in BAM files, it's in GFF files. It's sort of in a number of file formats. I guess just to sort of add some of the complexity and sort of diversity of the file types we're dealing with, people when they sort of are developing these file formats sort of in parallel to databases, people end up throwing a lot of the tricks and that we see in other places. So a SAM file format, which is used to store alignment information, has a number of fields which are tricky. So we have like this flag field, which basically is it's sort of like bitwise flags to represent if they're paired, if they're properly paired, if they're reverse strand, or like the first read in a pair. So you get this flag, which is an integer, which actually has, basically a bitwise representation. And then you have to use that representation to then say, like, okay, if I wanted to filter on all the alignments that are paired, what does that mean from going from the integer flag to the bitwise flags to the actual filtering, and do that in a way that is sort of meaningful to users. Then we also see another case, and I'll touch on again briefly later, of these tag fields, which are, again, somewhat like standardized, non-standardized ways of storing data, but have types and have values and have names. And so can sort of parse those, quote unquote schema on read, if you will. And then I won't touch on this much, but just want to mention it just because, we have, I think, potentially a number of students in here. And so, like, if you're getting ready for interviews or whatever, you might have to do like string alignments. Like that's a ubiquitous problem in bioinformatics, and it shows up in much more complicated ways too, especially for string searching, where so much of sort of functional genomics is really based off of, like, I found this sequence that I know does something. Let me find other sequences that do something similar. And in Exon, we have a sort of local alignment, but we don't yet have like an index-based search, which is what you see in this figure right down below, where it sort of goes through multiple pre-filter steps of like of searching on k-mers and then doing alignments. So some work still to be done there, but again, this, I guess, just an example of like a common workflow in bioinformatics, sequence alignment, sequence search, which is somewhat supported in Exon. Okay, so that was maybe like a high level of like the files. Now I want to talk a little bit more about sort of like how Exon does some of the actual like data processing. So so to add a file format, we have like FASTA, which I mentioned earlier, and really it sort of happens in a couple different steps, where we sort of get the raw file off the disk. We're then sort of building up these arrays. We're building up these batches. Then we're ultimately going to the streams of a record of arrow record, batches. And so, so I think this is very similar to some of the stuff that like Andrew Lamb would have talked a little bit about in his first talk of this series. So really sort of it starts building up and trying to handle the different like types of sequences you might deal with. So what we're looking at here is basically the lowest level, how we parse a sequence. And so there's a couple of different ways that are built into Exon to do this. So we support, of course, like strings like UTF-8, large UTF-8, like you end up needing a large UTF-8s to source some of the large genomes. And then also we actually have it built in, and I'll talk a little bit more about it sort of at the end, which is integer and DNA encoding. And so this wouldn't be a talk we didn't talk a little bit about sort of ML and AI, but the same way that folks have started using, say, like language models in all sorts of contexts, people are using language models for DNA and protein modeling now too. Really, it is just these sort of tokens of strings. So we're building up the sequences. We are then sort of building up these arrays, and then this is kind of what this looks like. This is a very traditional, like, sort of row-based to columnar-based file parsing. So we have this FASTA array builder, and more or less for all the file formats supported by Exon, we have something that looks very similar to this, where we'll sort of create an array builder, try to understand if we can push down any of the projections down, and then have some mechanism by which we can append to that array and kind of build up the batch. So this is your very classic, I'm sure folks have seen something like this before, where we're more or less just sort of transposing the data from the row-based to the column-based. And then finally, I know this might be a little tiny, but then we have ultimately have a batch reader. So what this is sort of responsible for, and again, this is sort of handled in multiple of the different types of a file format, but we have this batch reader, and we can basically read a batch, and all it's doing is taking the record off of the disk and appending it to the array builder. Does that a number of times, and then that's where you get your batch. And so that's kind of the standard, somewhat uninteresting, I guess, mechanism by which we're taking these row-based file formats, turning them into sort of columnar-based stores, so then we can use the standard data fusion infrastructure. And then finally, and this gets a little bit to the sort of trait-based extension of data fusion. So data fusion has this file opener mechanism, which is a trait. And really, so we basically just have a FASTA opener, which knows how to get the FASTA file. So one of the, I haven't talked a little bit too much about this, but object stores are ubiquitous in bioinformatics. Basically, there's so much data that nobody really could store this on a hard disk drive. And so people will store these on S3 or GCS or whatever you're using. And so so we get the result, convert that into a stream reader. We definitely support different compression types. I just wanted to make this a little bit shorter for the screenshot. Turn this into a stream reader, a stream of batches, and then, this is sort of it for the table provider for a FASTA. So as soon as this is implemented and added to Exon, you can do all sorts of queries as necessary to say, select all from. You just want the IDs. You then get the sort of predicate pushdown I mentioned, or excuse me, the projection pushdown I mentioned. And you get basically everything you could want. Well, not everything, but the 99% of SQL that you need to do SQL stuff, you can do with this. And then so once you have that, you can then start doing really cool analysis. And this is sort of a real use case here, where we are using that FASTA table in conjunction with the GFF file. So we have a GFF, which is sort of CRISPR. People probably have heard of CRISPR and Cas9. And so the idea here is basically you have a GFF file. You can then have a FASTA file. You can join the CRISPR where you have the CRISPR annotation to the repeat unit. So basically, within CRISPR, without getting too much into it, there are multiple parts of CRISPR system, and so we can join that, and we can join that to our FASTA and select only out the repeat unit. So this is a workflow that traditionally would have had to have been done by basically somebody cobbling together CLI scripts. But now something like Exon, you can do a lot of this again, sort of this tertiary analysis where you're joining data together and pushing it back out eventually to the FASTA that you have at the end. So,
and Trent, do the users write this query, or is it generated by a visual tool that lets them query this? Is it a dashboard or something else that drives the query?
So they would write this query themselves. You definitely. So it's one of the hopes, and there's maybe an interesting discussion for some. What's the best way to say this? I would love better interoperability, say with BI tools or other tools which maybe have a more GUI based interface. I think one of the challenges right now, at least with what I have found, is those tools don't have great capability, for example, to read ADBC, or the other route is maybe to sort of go over the Postgres wire, but that's kind of complicated for other perspectives, I found. So yeah, this is somewhat a user is writing this. What they actually end up doing, and I'll talk a little bit more about this later, is on top of Exon, there's a tool called BioBear. And BioBear is actually like a Python API, which you can use these. You can basically, it's like kind of like Data Fusion Python, but has all the Exon capabilities into it. So you would write this query or write some other query similar to this, get like a Polaris or a Pandas data frame back out, and do something you needed with that. So I'm definitely writing the SQL here still. But I would love better interop with BI tools that are out there, but I haven't, they don't seem to support that really well yet, unfortunately.
Thank you.
Yeah, thanks for the question.
Quick question.
Yes.
So far, I think what you've talked about is kind of like how to do the data modeling for the files, that the scientists are using. I'm just curious, like, will you talk about, or have you considered like, how to kind of bring their domain specific types to kind of be like first class citizens in the database, right? So, when you have a FASTA, like, do you do anything about like the sequence region is for these genes, right? Or like, like in this SQL query, even there's like a scan from GFF where type is CRISPR, right? So like maybe there's something that's somehow like, CRISPR specific that is maybe built into an interface or lower in the actual system.
Yeah, no, so that's a great question, and I'll talk a little bit more about that at the end. Like, so I talked a little about sort of like primary, secondary, and tertiary, and sort of right now I would say Exon exists very much at like the sort of tertiary level where we can do a lot of the tips or a lot of the fancy stuff to read the files, but there is not sort of like, say, moving up to more secondary analysis, which is kind of where you would say get this GFF from, that then has like the specific type that say is CRISPR. So outside of Exon, you could potentially like index the GFF and then have fast predicate pushdown to only get the CRISPR types. But there's not sort of like, if I'm sort of understanding your kind of your question / point better or well enough, sort of like the higher level, like, okay, I have a FASTA. I want to say generate the annotations on that FASTA, or and specifically maybe be able to run like certain types of tools or annotation generating tools, and to say, give me the CRISPR annotations from a file. So so it's very much kind of like the tertiary level of sort of joining. I'd very much like to get it sort of pushed down to sort of the secondary layer of analysis at some point, but it's not really like within Exon's capabilities yet.
Okay, thanks.
Mm-hmm. And then I guess I wanted to talk a little bit. So if we come back here, sorry, I'll just sort of speed up a little bit. If we come back here, we sort of see at this very end where we can actually like copy CRISPR to CRISPR.FASTA. So actually like copying file back out into the file format. That turned out to be like a little bit trickier than I had anticipated. And really it involves kind of understanding and injecting some Exon capabilities into really the sort of like three, very rough three components of sort of like parsing the underlying SQL, representing the logical plan, and then actually like doing the physical execution. So what we're seeing on the right is basically just an Exon specific parser it's wrapped around the Data Fusion parser. So this is very much sort of like the idea of like, okay, we have Data Fusion has a really good parser for 95% of SQL. But if we want to start doing some other interesting stuff, say like storing our file types as an Exon capable file format, we then need to sort of wrap this in wrap Data Fusion's parser in our own, say like Exon parser, which can produce then two types of statements. We have an Exon statement, which is an enum for a Data Fusion statement, and an Exon copy to statement. And so we then sort of build off that where we have the copy to, and then we actually turn that into sort of a logical plan where like there's an Exon logical plan that has an Exon data sync logical plan node. And then inside of that, we sort of extend to the data fusion query planner and the sort of physical planner to then know how to take that data sync logical plan node and turn that into actually like writing bytes to the disk. And so really like this writing the file formats, I think, was kind of involved, but really sort of was able to show some of the nice capabilities of data fusion where you can really extend more or less like all the parts of the query from the SQL itself to then the physical planning and ultimately the execution.
Was there like an existing implementation that did something similar that you used as a reference point, or is this you just winging it as you went along?
It was a bit winging it, and actually, I wrote and I got some feedback from Andrew Lamb because I made the data fusion parser actually wasn't public when I first started working on this, and so part of it was making that available to everybody from data fusion, and then kind of understand I guess winging it more or less.
Got it. Thanks.
So really, I think I thought that was particularly interesting. So that's sort of custom file formats. I then want to talk a little bit about sort of like the indexed IO. So one of the really, I think, great things that Data Fusion has is something it's called like a listing table provider. And so, like I mentioned, so much of bioinformatics is a file-based file format or a file-based science, if you will. And really what that ends up involving is, especially sort of the mechanism or the way people tend to store data is often say like each chromosome might be different files. So you might have like the entire human genome or somebody's genome that you want to query. And but what it actually looks like in practice is say 23 different files, each for like a different chromosome. And so you really want to be able to treat those as one like contiguous table. But you also want to be able to say do like predicate pushdowns to be able to search relative to like the sort of indexing that I described earlier. And so the way that we achieve that within Data Fusion is folks who work with Data Fusion, there's this notion of sort of like a partitioned file, which is sort of like a location in an object store. But then you can sort of attach this additional metadata to them. So when we, during query planning, get a query to a table, which is a set of VCF files that represent sort of like one underlying genome, what we do is basically parse that with the predicates that they may want to search, say here, chromosome 2, position 1 to 3, chromosome 2, position 4 to 5, and generate partition files for the each region that we want to subsequently filter. And so we can make these queries to say object stores, both like local file and object stores like S3, and do so in parallel. And so there's some users and there's some academic users who use this and who get like a lot better querying performance even relative to some of the sort of traditional CLI tools, because we're able to really, like when I say we, it's basically take advantage of the Data Fusion infrastructure to generate this data and query this data in parallel, which is great. And so, just sort of kind of an example where, we sort of take these, take these locations, generate the sort of like the file with location, and then ultimately look up the bytes and then do the sort of like byte selection that I mentioned earlier, just right here. Okay, so then we're sort of like when we actually do the physical execution, we're looking at the byte ranges and then looking in the actual files. Okay. Okay, now I want to talk a little bit about schema on read, so to speak. It's kind of like tongue in cheek a little bit, but so this is for SAM, and this is basically Rust like in its in its glory. Which is, if you've worked in Data Fusion at all, or sort of these other like database systems on top of or sort of in Rust, you basically end up seeing these like gigantic match statements. And that's a lot of what this is in here. And so like, for example, we have this tags and data type from the data. So this data is basically the tag that I mentioned earlier. It's coming out of SAM, which is basically like a tag, a file type, and the actual value. And what we're doing is basically looping through this data and more or less like lots of matches, right? So when we're constructing the schema here, we're sort of matching. Okay, so we have a character, a string, or a hex, and what does that mean? We have an integer, yada yada yada. We then have there's even like array types. So this is the sort of schema generation. So when you run a query, it does like a quick basically sniff of the SAM file, parses this data for it, generates a struct field at schema creation time. And then on the right side is where you actually see us sort of like building up the tags, and the very like similar process is happening there, where you get the sort of tag information, is if it's none or if it's some, and then you work through these giant enums, right? So it's you basically, yeah, I'm sure folks have seen these massive enums before in lots of contexts, but especially database systems in Rust. The other thing I wanted to mention is we take pretty big advantage of Data Fusion's UDFs. So I talked a little bit earlier about the sort of binary flags that show up in the flagged field, and the way we do that in Exon is we have these scalar functions where you can run the scalar function on that flag. Say is segmented, is properly segmented, is unmapped, is mate unmapped, is reverse complemented, and so forth. And we have basically a whole slew of them that align to the different flags as they show up. So on the left, you basically see the sort of very generic SAM scalar UDF, which again is just like a UDF in Data Fusion, it basically just maps on a particular type of flag or a particular sort of like setting of a given flag, and makes that available. So it makes it pretty easy for folks to not have to sort of deal with some of the nuance and the sort of minutia, if you will, of these flags, and can just sort of run them in kind of generic or human language, if you will. Another area where we take advantage of sequence search, or this is sort of like the local alignment I mentioned, and nothing too exciting here, but basically it's kind of when I was able to do this was kind of when I started getting like really excited in Exon, but more or less to build up a scalar UDF that allows folks to run alignments. So much of bioinformatics is saying like, does the sequence look like this other sequence, which is really like, how does it align? Where does it align? And some of the capabilities you can do with Exon allow you to do that pretty easily. I should say, and this actually gets the sort of history of Exon. The first version of Exon was actually a DuckDB extension, and for various reasons, sort of moved away from that. But one of the things I think is kind of interesting is I did see some of the sort of nuances between DuckDB's DuckDB sort of UTF paradigm, if you will, and Data Fusion's UDF paradigm, in terms of particularly like DuckDB has a sort of setup and tear down mechanism that Data Fusion I think lacks to my knowledge. And this is sort of important due to sort of the aligner allocation. You basically build up this really big matrix that you use for scoring when you do aligning. Okay, gonna just touch on a couple things here with respect to like this is like database stuff and kind of on the back end. They want to touch briefly on since Exon is a Rust-based built on Data Fusion, your typical bioinformatician like knows zero Rust, and would not really be able to use it. There would be like a CLI available, but this is kind of what I was referring to earlier, where It's actually really easy, thankfully, just to like lift this up into other languages. And so the majority of people who use Exon, they actually use it via this BioBear package. So you got this really nice Python. You can do pip install BioBear, and you can then just run SQL, get Polaris data frames out, and go to town. And you can see, from a performance standpoint, I think this is actually quite old. So excuse me. I suspect we're actually much faster than this at this point, but you can see, people are working with multiple files where we're kind of on the 1/5 ish of the sort of time that it's been traditionally needed to work with some of these file formats in Python relative to the kind of tools that exist out there. And it's much easier, for folks who have done some work with sort of making Rust available via Python. It turns out to be a very, very nice experience. We also have one of our heavy users that uses Exon via R, and so another area where you kind of use data fusion and Rust and its sort of capabilities to be extended out into other languages. And so, so it becomes very idiomatic in R, but you get all the speed and niceness as a developer in Rust. So I just want to touch briefly as sort of like the last section on sort of making Exon better and kind of what that looks like in the future. Spatial transcriptomics is a huge area in biology, and it's sort of only growing. What I've talked mostly about thus far has been really on sort of like NGS and NGS adjacent analysis, but there's sort of growing work to do to add, again, sort of the spatial modality into biology and bioinformatics. And so there's this really exciting package, AeroZar, which has some data fusion capabilities that I actually added. And ZAR is a file format for ND arrays. For example, you might want to select some area of like an organelle based on a bounding box. So it's basically geospatial and sort of spatial transcriptomics are where we see the sort of AeroZar format, but it does have the capabilities of being used with data fusion, which I think is exciting. Yes, this gets a little bit about how the sort of desire to sort of move up into from tertiary to secondary, again, sort of kind of being AI ready a little bit. So many annotation methods are AI or ML. This has been going on since like the 70s and 80s. So like ML wasn't as like really called that then, but so many of the statistical models in bioinformatics are based off HMMs or really like the sequence-based analysis. And so, but that does introduce some complexities, both with respect to hardware, GPUs, obviously, and sort of artifact management, where you have, say, some you trained a model, you trained an HMM, you did some amount of like model training, then you need to get your artifact to work with. And so we don't do much of that yet, but we do have some capabilities around just straight sort of integer encoding. And so, like, for example, in Exon, when you're parsing a FASTA file, you can actually set an option to integer encode DNA. And so rather than sequence being like a UTF-8 file format, it'll be an array of integers. And then sort of how this looks like a little bit in data fusion. Data fusion actually has the capability of adding sort of like function factory, where you can add, sort of have functions very similar to what you might see in like Postgres, where you can do some sort of artifact management. And this is sort of PSSM, which is a scoring matrix. So we've done some light work here, but hopefully do more in the future. And then I guess finally, just a little bit on sort of like better search and alignment. So talk a little bit about sort of local alignment, but folks make indexes all the time in databases, but really that shows up all over the place in bioinformatics too. And I would really like to add sort of capabilities where you have a set of FASTA, you can generate MMSeqs index, for example, and then run queries on that index. And I think that would be great from a user perspective, where what you see and what you tend to have to do right now is very much in this lower left-hand part, where you're, again, this very sort of like command line CLI based work. So that I just want to kind of wrap up and touch on a couple of things real quickly, which is to say, right now Exon is very focused on the sort of tertiary analytics and sort of analysis. It does some of those things really quite well and better than the sort of like tools out there. I didn't really include any benchmarks, so you can take my word for it or not. But we've sort of seen from users and benchmarks that I do have that it's on par or faster than a lot of the existing tools. One of the main challenges I would just mention, I think this is maybe different than just gets a little bit to some of the questions, which I think are right on point, which is building a domain-specific database is it's great. One of the challenges that we've sort of come across in getting like user adoption is it definitely requires a sort of like it's like a sliver of the population in bioinformatics that can really make use of this right now. Like we have a couple of folks who do really good work with it and like tend to use it quite a bit. But it does require some sort of specialized, well, it requires the domain knowledge. And in this talk, it was like bioinformatics, but really if you wanted to apply databases to any other domain, it would sort of like require that knowledge and SQL. And so like though having those two skill sets in abundance is certainly growing, but it's sort of not all over the place. And that's, I think, kind of a key learning that I've had over the past year and a half or so of really developing Exon is the user base. If they get it, and they're like very productive with it. But it's not a huge group of folks just because of those two specialized knowledge or some of the specialized knowledge, which is why I think the question around making it more accessible is a really good and important one. And with that, I think I will leave it there.
Awesome. I will clap on behalf of everyone else. Trent, thank you so much for doing this. We have time for a few questions. If you want to go for it, unmute yourself and fire away.
Yeah, I've got a couple of questions, Trent. I think it also mirrors some things that were asked in the chat. How big are some of these databases? How large do they get? And do you ever have to worry about concurrent users? Or how do you update the database when you get a new set of information that comes in?
Yeah, so great questions. Right now, you might look at, so say like one genome might be in like the hundreds of gigs, or potentially more. So they can end up being like quite big files. If you look at like the 100,000 genomes project, I don't know how big that is in totality, but you might imagine that sort of times every genome, say a couple thousand genomes, you end up with like quite a big amount of data. The updated question, I think it's really an interesting one, and one that has kind of skirted around due to the, I guess I would say, like tertiary and sort of like workflow nature of the database, which is a lot of it sort of takes the output of those files as sort of like a given. Like there really isn't a notion of, say, like I want to insert a row into a VCF file. It's more like I had this VCF file that was generated due to some workflow. I then am going to query that down to some subset based on my specific question. And then I will output that to, say, some other file format to continue on, or some chunk of IDs, whatever it may be, that's sort of germane to the task at hand. So the updating really doesn't come into play much, because it is most of the time treated as like kind of read only, if you will, where it's like the output of some file or some bioinformatic process. You do some sort of like further selection on that and then work with that subset of data versus doing something and then wanting to, say, like insert data back into a specific file.
And what kind of end analysis are typically the users carrying out? Obviously, the genome alignment, whether it is de novo or something else, is done outside, right? You are basically storing things that are probably SNPs, so a lot of SNP analysis and stuff like that? Or what's the type of analysis people are doing from the user perspective? And for the domain?
Yeah. So I mean, it depends a little bit on like what like what the sort of like sub area is so to speak. So like one area quite a bit is like say searching for proteins where you really have like a protein database. You have some set of annotations that you generated on that. Say like protein families, for example. And that is then where you have, and if you go back to sort of file types, where you have, say, like the FASTA file of the proteins, and you might have like a GFF file or a TSV with the PFAMs called on those sequences, and then you want to sort of join that data back together. A lot of it is just sort of simply using some of the capabilities that Exon has to work with indexes to filter data down like really quickly, then to get it into Python and a Pandas data frame or a Polaris data frame. So a lot of that is sort of saying like, okay, I have this massive data set with, tens, hundreds of files. I know I need a specific region, and just can I sort of like use Exon to really, in that case, use BioBear to sort of turn that off or run that very quickly and run those sort of scans, but really, again, because we sort of take into consideration the index information, we end up not having to scan a lot of data, so we can do it very quickly. And so a lot of it is like, I know I need this subset of data out of this very large data set. How can I get that very quickly? And so that's where like Exon comes into play and gets a lot of its uses. How do I get this data in Python, basically?
I have one more question if no one else has. Are you seeing a whole bunch of new data coming in that is synthetically generated, whether it is from AlphaFold for protein structure data that someone's trying to integrate, or has the world changed with so much synthetic data generation in the bio world?
That's a great question. So I've done like a bit of work on sort of the AlphaFold. AlphaFold right now, it definitely becoming more and more popular, and folks are just like having programs where they're just like generating structures, kind of to your point, for a ton of proteins. It still is arguably a little bit too low throughput to do some of these sort of like initial screens. So you sort of imagine where you might have like, say, 1 billion sequences or like 10 billion sequences. Where sort of Exon kind of fits in is how do you go from that to say like 1,000 sequences or like 5,000 sequences, and then that's where like AlphaFold might come in after that, is where you want to generate the structures of those. So you definitely see it more and more use, and like 5 years from now, like the answer to your question might be very different. At least what I've seen so far is AlphaFold is great, but it's still slightly too low a throughput to just sort of turn it loose relative to sort of entire sequence data sets.
Cool. Thank you.
Yeah, thanks for your questions.
One last question in the chat. Do you think that data fusion is well suited to ingesting, analyzing data represented as multidimensional arrays, data that's typically stored as HDF5?
I think that's still TBD. Some of the work on sort of like ArrowZar definitely makes that interesting, and so I'm excited to see where it goes. I have not done enough with it to maybe give a complete answer, but definitely seeing that as a very interesting opportunity, especially as you go from, say, like the tertiary analysis, again, just coming back to that briefly, where you might want to sub-select down your data based on a spatial query and then join that to other additional data. So it's definitely an interesting avenue. I think it remains to be seen if data fusion's and data fusion's sort of right solution there, but certainly has the you can certainly extend it in very like in many ways. So it's certainly an avenue worth pursuing. Just maybe a bit too early to say concretely yet.